Yeah. Okay, I think we can start. So let me just present my screen. Okay, so for today, we're going to be talking about nucleic acids, then the central dogma, and then the protein synthesis, or the last part is the protein synthesis. Okay, okay. So your nucleic acids are actually made up of nucleotides. Now, this is the monomer, and at the same time, you can actually find it in the DNA and RNA. Okay, so you have there, it carries the genetic information, okay, and it can be found in the ribosomes, chromosomes, and chloroplast. Okay, so that's so your nucleotide. Now let's go ahead and examine the different characteristics of your DNA. So let's us first look at the different structures inside. So the first one is the bases. Okay. So your bases, we have two types of bases. We have the pyrimidine. Okay. Let me just put on my printer. Yeah. So you have the pyrimidine and the purine. Okay. So let's concentrate first on pyrimidine. So your pyrimidine is a heterocyclic aromatic organic compound. So the reason why it's heterocyclic, if you look at the structure here, one of the component has nitrogen, two nitrogens inside its cyclic structure. Pag sinabi kasi natin homocyclic, it only contains carbon. Okay. Now, it actually forms your cytosine, thymine, and uracil. Okay. Now, the other one is your purines. Now, your purines is a double ring. It's actually composed of a pyrimidine ring and the imidazole ring. So, the imidazole is this part. Ito. Okay, and it's also heterocyclic. Given that it has a pyrimidine, dalawang nitrogen na doon, and the imidazole, dalawang nitrogen din na nakakabit. Then it makes up your adenine and guanine. Now, your adenine, ang kagkakaiba nila, if you're going to look at the structure, is that for the adenine, you have one ammonia group or amino group in it. And then for the guanine, you have one amino and then also one carboxyl on top. Okay? So, yan. Now, take note that these bases, the pyrimidines and the purines, have what we call a pairing. Okay? So, it's usually thymine or uracil. Usually, it's thymine when it's in the DNA, and it becomes uracil when it is in the RNA during transcription. Okay? And then it has two bonds there, and then for cytosine you have there three hydrogen bonds. Now take note, both of them are hydrogen bonds. Okay, then the next structure is the sugar units. Now take note that both of them has ribose. Now the only thing that's different about them is that on the second carbon here for the DNA, it is deoxyribose. So it means it's no longer containing oxygen. It's only having hydrogen. And then on the ribose, you have the complete hydroxide attachment. Okay? So this is now your DNA is deoxyribonucleic acid, and for RNA is ribonucleic acid. Now, the last portion of the structure is the phosphate bond. Okay, so it's actually familiar. We've discussed this with bioenergetics, the ATP. Although here, the one that you're looking at, hindi ko na-specify si adenine dito. General formula lang siya. Okay, and if you will notice here, you have here the base plus sugar. You call it as a nucleoside plus one phosphate group. It's now called as nucleoside monophosphate. And then pag dalawa yung phosphate, it's nucleoside diphosphate. And tatlo, nucleoside triphosphate. Okay. Now take note, as we've discussed in bioenergetic, the energy is coming from the phosphodiester bond that is occurring between the phosphate groups and the sugar molecules. Okay. So yun yung nagre-release ng energy natin that is needed for the reactions. Okay. Now here is the table showing the different names of your bases along with the sugars and with the sugars. Okay, so you have your nucleosides and then the nucleotide. Okay, so if you will notice, if it is for the nucleoside, for the purines, it's ending with sine, while for pyrimidines, it's ending with dine, D-I-N-E. Diba? Adenosine, guanosine, deoxyadenosine, deoxyguanosine, while for pyrimidines, it's cytidine, timidine. Okay? Parang timid-timid. Next is for the nucleotides, you have adenylate. All of them are ending with ATE. Okay? So just showing you where it is usually found. Okay? So again, adenine, guanine, cytosine are all found in DNA. Thymine is found in the DNA. Uracil is in the RNA. Okay? So let's go now to the different functions. So the basic, besides the basic function of the DNA or the nucleotide is to carry the genetic material, it also has functions, diba? One of them is the adenosine triphosphate, which is the major source of energy natin. Okay, and then at the same time, they also function as coenzyme like NAD and NADP. This is actually derived from ADP, adenosine diphosphate. So that's why it's also used. And then at the same time, they can also function as messenger molecule. You have there the adenosine monophosphate. Now it's in the cyclic form, therefore actually functions as a regulation, metabolism, and transporting chemical signals. Okay. Now, importance of the DNA, take note that it contains the hereditary molecule that maintains and transmits the information of the cell needed for it to survive and create offsprings. Diba? Kaya nga, diba yung... Oh, wait lang guys. Okay. So the functions of the DNA is to replicate itself during cell division. Second is to direct the transcription, which is actually also known as the creation of the RNA. And the information that it contains is found in genes, which are sections of the DNA molecule that contains a code, and that usually the cell uses to create RNA. And finally, the proteins. This is actually known as the central dogma. We will expound on that later on. Now, take note the characteristic of the DNA that it is a double-stranded helix, and this structure helps store information safely by essentially maintaining a double copy of its information. Okay. Now, in terms of RNA, RNA is created when the cell reads the genes from DNA and makes a copy of them. You might say that the RNA is also known as the working copy of the DNA. Now, during transcription, it's actually producing three types of RNA. You have the mRNA, which is it is the working copy of the DNA and brings the cell's machinery to create the protein in the ribosomes. Okay. Next is the ribosomal RNA. So this is actually the ribosomes uses the information in RNA as a blueprint to create the proteins in carry out the nearly all of the cell's function. And the last one is the transfer RNA, which carries the amino acid to the ribosomes in order to synthesize proteins. Okay. Now, in terms of structure, we have actually three levels. So the first one is a primary structure. Okay, so you have here your phosphodiester linkages. Now, this is the name of the bond that is formed between phosphate and the carbon or with the sugar. Okay, that is the link that is happening. Okay, now take note the direction of the DNA is usually from the 5 prime to the 3 prime end. So may say 5 prime, ito yung sa ditong part ng DNA or ng mismo sugar molecule. Yan, this is actually the fifth carbon. And then here is the other side naman is um, 3 carbon. Okay, so 5 carbon here and then 3 carbon kasi ito po yung C4. So ito po yun, dito siya nakakabit. Okay. Okay, now inside the cells, the nucleic acid synthesis occurs by forming new phosphodiester linkages at the 3' end of the growing polymer. Okay? So yan po yung kanyang movement. Okay. Now, since it's since it's actually forming a link between the phosphodiester linkage between the phosphate and the sugar molecule, therefore it's actually allowing the bases to be exposed to bond with other bases. Okay? So ganyan po yung kanyang nangyayari. So this actually creates now the different um, nitrogenous bases sequences. Okay, here is another structure showing the sequence drawing from 5 prime to 3 prime end. Okay, so this is actually a shorthand notation of the sequence. So that's ACGTA. Okay, so yan. Now, the secondary structure of the DNA is called the double helix. Okay, so the double helix is actually a anti-parallel. Take note, why is it anti-parallel? Remember, I mentioned it's a 3 prime a 5 prime to 3 prime. So the opposite side is going the different direction, okay? But it is wound around in the same axis. So the sugar phosphate chains wrap around the periphery. So yun yung nagpo-protect dun sa bases inside, okay? And then ando dun yung kanilang complementary bases. Now this is actually na-identify to ni na Watson and Crick when they were able to get the structure of the DNA. Now, take note that the type of bond that is occurring in between the double helix is hydrogen bond. Okay, so you have there a partially positive hydrogen from the amino group and the partially negative oxygen from the carbonyl group, causing them to form now your hydrogen bond. Okay. By the way, guys, take note, it's still recorded. I'll upload this later on. Okay. So yan. Now, inside the base pairing here, take note of the characteristics. Marami po tayong klaseng na DNA na na-record. You have the alpha, the beta, and the gamma. But the most common is the beta DNA. Ito yung common existing sa nature. So here, if you will notice, we did mention earlier that for adenine and thymine, it is double bond. And then for cytosine and guanine is triple bond. Now, if you go back to our organic chemistry, you will remember that we mentioned that if it's a double bond, comparing it with a triple bond, yung triple bond, it's causing the molecules to become closer to each other. While for double bonds, medyo relax konti siya. Okay, and this is an example of that one. If you're going to look at the diameter or the distance rather between carbon number one and carbon number one from the other bases, for the cytosine and guanine, it's actually 10.8 angstroms. This is actually a little bit smaller compared to the adenine and guanine, which is 11.1 angstrom. Although it's just 0.3 angstrom, guys, yung itsura ng ladder nyo, parang nagko-curve siya. Parang mga ganun ka na sa TikTok, di ba? So, yun. So, parang ganun yung dating tuloy niya. Now, in order for it to become stable, nagkakaroon tayo ng what we call the helix formation. Okay? So, here, take note, another characteristics is what we call the base stacking. Yan. So, the bases in the DNA are planar and have a tendency to stack, and it forces the hydrophobic interaction and the van der Waals forces interaction. Now, this also causes it to form now your grooves. Okay, so this is what's happening for it to maintain its structure. Remember, pa-curve siya. Hindi pag-curve yung ano, di ba? Hindi masyadong, parang hagdanan yan, pag alanganin na itsura ng hagdanan mo, hindi masyado stable. So, kailangan niya mag-twist. Okay, so take note of the characteristic of its helix formation. It is right-handed. Okay, and then at the same time, then the distance between the base pairs is at 3.4 angstrom. Now, don't worry guys, hindi ko na siya tinanong sa questions nyo sa final exam at saka sa quiz about dito. At least it's good to know about this one. Okay. Now, we have what we call the minor and the major grooves. Now, yung major grooves po, yung pagkakatwist niya is actually around 12 angstrom across. So, pag umikot siya, 12 angstrom yung difference niya. Tapos, another twist, yung minor groove, it's around 12 angstrom. So, ang laki yung diferensya nila. Okay? And the base pair is usually perpendicular to the helix at 3.4 angstroms apart. Ayan, nasabi ko na pala yan. Okay, so each turn of the helix is 36 angstrom with a plus or minus 10.4 for the base pairs. Okay, so again, with the minor and major groups, usually it's opposite of each other, and each runs continuously along the entire length of the DNA molecule. They arise from anti-parallel arrangement of the two backbone strands. Note that the grooves are actual structures, features of the molecule, not consequences of the way it is drawn. The grooves are important in the attachment of the DNA binding protein involved in the replication and transcription. So, ito po yung itsura ng mga itsura nila. Okay. And then here, yan. So, ito po yung pagtutwist nila. Okay. So, yan. Now, from here, to pag-twist nyo doon, that's your minor groove. And then, yung isa is yung major. Mas malaki, of course, yung major groove. Okay, now the tertiary structure, this is actually referring to the folding into chromatin. So usually your DNA forms now coiling around histones, and histone is a protein. Now, yung protein, yung DNA bound to the histones forms now the nucleosomes, which is around 10 nanometers fibers, and the nucleosomes contains 146 nucleotides. So, ito po yung itsura ng nating DNA na nagfo-fold or nagra-wrap around sa histones. Okay? Now, any of the repeating globular units of chromatin that is inside of the complex is made out of DNA and histone. Okay, so ito po yung kanyang itsura. Ito naman po yung actual structure niya on a molecular level. Okay, space filling. Okay, so yan po. Now, if you're going to look at the hierarchy or pagiging compact ng structure, ito po siya. So, you have your DNA, diba? You have your double helix, and then it's now forming beads of string to form the chromatin. Now, your chromatin forms now a fiber which actually forms a coil. Okay? Now, your coil forms now a rosette. Okay, nagka-coil sila dyan. And one coil is made up of, one loop forms, 6 loops forms 6 rosette, and then 30 rosette produces one coil. And 2 chromatids produces 10 coils. So if you're going to analyze or kung gusto niyo ma-visualize kung gaano kalaki yung DNA, so let's say meron kang isang ka-size ng hand mo yung isang DNA strand, mapupuno mo yung isang baseball field if you unwind the DNA. Ganun siya kalaki. So makita nyo na lang to, di ba? Ang liit-liit ng DNA, tapos yung chromatid, ang liit-liit din. You need a powerful microscope to look it up. Okay? Now, yung pagkaka-supercoil niya is actually similar to the supercoil that is happening on a telephone wire. I'm not sure if you've seen this. Yung nakakita pa naman siguro kayo ng mga ganitong klaseng telepono, di ba? Pag, alam mo yun, pag nagtitele, babad ka, tapos paikot-ikot ka, na tendency ng wire, nagko-coil siya. So, ganun din po yung pag-supercoil na nangyayari sa ating DNA. Okay? Para yung maprotektahan din yung kanyang information, yung mismong genetic material. Okay. So this time we're gonna be discussing about DNA replication. Okay. So usually you have there two strands of DNA na double helix are usually separated. So this is actually just a summary of what we will be going through the actual steps later on. Okay. So take note that the replication is what we considered as Tanto, this is considered as anti-parallel, yung kanyang structure, pero yung mismong process niya is what we call semi-conservative replication. Why semi-conservative? Because you maintain half of the material on the new. So half old, half new. Kaya ang tawag sa kanya is semi-conservative. Okay? So usually you have their enzymes that are involved in the DNA replication process, which are template-directed polymerases that can synthesize the complementary sequence of each strand with extraordinary fidelity. Okay, so so how is it copied? So you have here, so ito yung ano niya, overview pa rin yan. So you have here your DNA, maghihiwalay sila, and then the specific pairs will be created. Kumbaga yung ka-partner ng bawat component will be creating a new strand for it. Okay, kaya nga tinawag siya na semi-conservative. Half old, half new. Okay, so according nga kay Watson and Crick, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. Okay, so yan po. Now, let's go now to the steps. Okay, so, take note again, as mentioned, DNA is anti-parallel. So, ito yung sasabi ko sa inyo. Yung isang side is 5' going to 3'. Tapos yung isa naman from 3' to 5'. Although yung direction niya opposite. Okay? Yan. At the same time, yeah, it's replicated only at a 5' to 3'. So, yun yung direction niya. Eh, paano ma'am yung kabila? Later on, you will see that it's gonna be on a staggered replication. Now, although yung iba, hindi na mini-mention to, but I'd like to include as well the topoisomerase. So, the topoisomerase is actually the one that cuts the DNA at a particular point and it unravels the supercoiling. So, di ba, naka-supercoil yan, nakapulupot siya. Siya yung mag-uunat. Okay? So, pag naunat na siya, pwede na siyang mag-start ng replication. Usually, it will identify an adenine thymine rich location. Doon sila mag start ng replication. So, may dadaan dyan, tapos nakita, oh, may rami dito adenine thymine, cut, tapos unwind. Okay? Now, we have two types of topoisomerase. You have topoisomerase 1, which cuts it without any ATP utilization. Then, they have also the topoisomerase 2 that requires ATP. Okay? So, yan yung pagkakaiba nila. So, nag-unwide na tayo, di ba? So, the next step is zip na natin. Okay? Good morning. So, here, you will notice the DNA is now zipping out. Parang alam mo yun, yung para siyang zipper ng pantalon, ng jacket ninyo. So, ganyan yung nangyayari dyan. Okay? So, yan. So we have here now the unwinding of the parent DNA using the DNA helicase. Now here, it's usually accompanied by a couple of proteins. They are not enzymes, they are proteins that aids in the replication. Okay, so in some resources, take note that the DNA helicase moves at a 5' to 3' direction. So if you're going to look at the diagram here, yan, zoom in natin. So, dyan siya, 5' to 3'. So, yung iba, dinadrawing yung helicase na parang zipper. Yun yung parang pambukas ng zipper, yung dila na tinatawag natin sa zipper. Yung iba, nakakabit lang siya dito sa 5 to 3 prime, hindi nakakabit dito. Okay, so yun yung nangyayari sa ibang mga pictures. If you're going to Google it up, you will see na nandito siya sa 5, 3 prime nakakabit. Kasi yun yung direction niya eh. Kung dito, parang yan, parang style yan ng elevator. Gaganong-ganon yan pag nagkataon. So, kailangan doon sa kabilang side siya kasi yun ang direction ng punta niya. Okay, so one of the components here is the during the replication is DNA protein. So it actually binds to a specific nucleotide sequence at origin, causing a short tandemly arranged, and this is usually at the adenine and thymine. So siya yung nagkakaroon ng melt. Kaya nga may tawag na term sila na AT melt. Okay, so yun yung sinasabi ko sa kanina. Doon, pag nakita na yung AT region, doon mag-i-start. Okay, so you have your DNA protein. Now, the melting is ATP dependent. Okay, so kung walang ATP, it's not gonna start. Okay, next is the DNA helicase per se. Yan. So these enzymes bind on the SSDA, single-stranded DNA, near the replication fork, then move to the neighboring double-stranded region, forcing the strands apart, in effect unwinding the double helix. Helicase require energy provided by ATP. And the last one is the single-stranded DNA binding protein or yung SSB-DNA. Okay? The function of SSD-DNA is, di ba naka-unwind na siya? Okay, di ba parang ano din yan, yung mga velcro guys, di ba minsan pag velcro, pag binuksan mo, pwede siya magdikit, ganun din yung tendency niya. Pwede magdikit yan. So what the SSB-DNA is that it would maintain them to be apart and at the same time, para hindi rin po masira yung mismong exposed na DNA, okay? Because they can disintegrate also. Yan. So, ganyan po yan. So, parang binuksan nyo, tapos meron nakakapit bale dun sa dalawang gilid ng zipper nyo. Okay? So, yan. So, let's go on to the next one. So, ito naman ang gagamitin nating diagram. So, hindi na natin nakita si topoisomerase kasi nandoon na siya sa harapan. You have your helicase na nag-unwind. Okay? And now, let's go to the next step. So, the next one is the RNA primase. So, this is actually an enzyme that adds a short segment of RNA on the exposed DNA. Now, yung part na yon na idinagdag is what we call an RNA primer. Kumbaga parang initiator lang siya. So, once it's already acting as an initiator, the tendency now is that it will allow the partnering ng mga complementary base pairs. Okay? So, take note that this RNA primer will not stay there. It's not gonna stay there. It's actually going to be removed later on. Okay? Kasi hindi naman siya part ng DNA. Okay, wait lang guys ha. Saan na yung mouse ko? Ayun. Sorry naman. Okay, going back here. So, yan. So, ito po yung position ng dalawang RNA primer. Now, bakit hindi natin nakikita sa kabila? Okay, you might be wondering, Ma'am, ba't wala sa kabila? Bakit nandito lang? Kasi po, since it's moving along the replication fork and the direction is following the helicase, wala siyang problema. So, malamang yung RNA primer niya nandun sa kabilang side, hindi na natin nakikita. Okay, tapos nag-continue na siya, nag-pairing na sila, and they're already producing now the actual DNA strand na. Okay, so may panibagong DNA strand yan. Okay, the next one is what we call the DNA polymerase. So ito yon. So dalawa yan. Actually, meron pa isa dito, yung kulay blue na yan. So this is actually the one responsible to add the necessary nucleotides to the new strand in the 5 to 3 prime direction. So dapat dun siya sa direction ng DNA mag-form. However, dito hindi po gano'n nangyayari. So what would happen is that it's gonna be magiging staggered, putol-putol tuloy yung creation niya ng pagre-replicate. So magre-replicate siya ng gano'n since nag-meet na siya do'n sa kabila, titigil na siya. So mangyayari ngayon, fragmented yung inyong DNA dito sa particular part na 'to. And this fragmented part of your DNA being produced is what we call the Okazaki structure or the Okazaki fragments. Okay? So yan po yan. Now, ito po kasi kaya siya hindi na natin nakita yung RNA primer dyan, tapos nag-coiling na kaagad siya because this is what we call the leading strands. So usually, the leading strands are replicated in one continuous space. Kumbaga parang dire-diretso siya, parang sumusunod siya sa trend. Okay? While on the other hand, the lagging strand here is na, yan yung opposite direction. Now, since opposite direction, napuputol yung inyong mga structures. Therefore, you are forming now the Okazaki. Uh, okay, wait lang. The Okazaki fragments. Okay, so this is one Okazaki fragment, another Okazaki fragment. So pag umabot na siya doon, oy, may RNA primer, hindi na siya dyan, lipat na naman siya doon sa kabilang side na available for sequencing. Okay. Now, once it's already produced, so what will happen is that you have another protein that will actually, or an enzyme that will actually check. Yung enzyme is an exonuclease, hindi ko na pala naisama dyan. It's an exonuclease that will actually remove yung mga RNA primer. So tatanggalin yan dyan. Okay? Tapos, papasok ulit si DNA polymerase para palitan yung RNA primer ng actual na base pair. Then you have now your DNA ligase that will actually repair or bond those fragmented Okazaki fragments to produce your DNA, your daughter DNA. Okay? So usually, may magpo-proofread pa yan eh. Checkin nila kung tama, baka meron pang naiwan RNA primer, tatanggalin, sipain paalis, pasok na yung panibagong base pair. Okay, so you're gonna be having now two complete DNA strands, which one is half old, half new. Okay, any questions so far, guys?
None
None?
po, ma'am.
Okay. So let's continue. So this time we're gonna go to the central dogma. So yeah, we started already with the replication. So this time we're gonna be going to transcription and then translation. So the central dogma is actually the process in which the instructions on the DNA is converted to functional proteins. And this is actually proposed by Francis Crick when he discovered the structure of DNA. Okay, so it's actually three parts. So it's the DNA to create another DNA is called replication, DNA to RNA transcription, RNA to protein is translation. Now, at the same time, we can also do a reverse transcription. Di ba? Remember the case of COVID-19? You are getting the bacteria or the virus, gagamitin yung mitochondria ng virus to generate the DNA. O kaya yung pag ano din, pag yung para ma-confirm kung tatay mo nga yung tatay mo. 'Di ba? Gano'n. Kukuha sila ng dugo mo. Remember, yung dugo, it contains mitochondria, you have there the cells, yun yung i-generate. Yun yung gagamitin para mag-produce ng DNA. Okay? So, yan po yung reverse transcription. Okay? Next natin is, yan. So, as I've been mentioning earlier, this is the DNA alone cannot tell the cells how to make proteins. It usually needs the RNA and other materials to produce the proteins. Okay, so take note that the DNA is located in the nucleus, However, the proteins are outside. It's in the cytoplasm. Sabihin nyo, ma'am, nasa ribosome, nakakapit sila sa ribosomes, ay sa endoplasmic reticulum. Yes, nakakapit nga sila sa endoplasmic reticulum, but it's still outside the nucleus, right? So, in order for the information to go out, it has to change to RNA. Now, ano yung pagkakaiba ng dalawa? So, we already mentioned that one of the sugar molecules is deoxy, the other one is um, uh, ribose, deoxyribose and ribose. Now, aside from that one, it contains one nucleotide chain as far as RNA, while DNA contains two. Okay, then yung base na RNA is uracil, thymine is for the DNA. Yan, ito ulit yung sugar. Okay, so here are the different types of RNA. So here you have the mRNA. So this is the copies the genetic instructions from DNA in the nucleus and carries out to the instructions to the cytoplasm. You have your rRNA. Yeah, it's actually the organelle where the protein is going to be assembled. And then you have the tRNA, which is actually going to bring the amino acid that is being called by the mRNA. Now, let us examine the parts of your tRNA. So, if you're going to look at it, it's actually a part of yung mRNA. Pag ginawa yan, hahatiin yan into the different parts. One of them becomes the mRNA, the other one becomes the tRNA. Now, yung tRNA will have pairing within itself. Magbabond siya within itself. And it's actually going to have three anticodons. Okay. If you're going to look here, you have the anticodons. Tapos yung sa mRNA is what we call the codon. So this is actually a three-letter base pair, and it will call now its complementary base pair also in threes, and you call that as an anticodon. Now, each codon is equivalent to one amino acid. Okay, so yun yung nakalagay sa labas. Okay, so yan. Later on, we're gonna look into that one. Okay, so let's talk about the transcription. So take note that the transcription happens inside the nucleus. Okay, so it uses DNA as a template to make the RNA molecule. Now, the RNA leaves the nucleus and goes to the ribosome in the cytoplasm where translation occurs, and the translation reads the genetic code in the mRNA and makes a protein. Okay, so let's go to the different steps. Okay, so unang-una is, there are actually three steps. So you have only initiation, elongation, and then termination. Now, in the initiation stage, you have here an RNA polymerase that usually goes over the DNA. So, iikot yan. Dadaan siya doon. Okay, hanap, 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 hanap. Hahanapin nila yung what they call the promoter region. So, pag umabot siya doon, ayan yung promoter, start na yung polymerase. And what the polymerase does is it opens your DNA. Hindi siya yung katulad ng helicase. I-open lang siya para magkaroon ng transcription. Okay? So, yan. So, the next one is what we call the elongation. So, habang binabaybay, habang binabasa ng RNA polymerase, yung mismo DNA natin, binabasa niya yung complementary na pang-RNA. Okay, so pag nabasa niya ngayon, habang dumadaan siya doon, it will produce now your RNA as you can see here on the slide. Okay? So yan, gumagawa na siya ng RNA. Then, aabot siya ngayon dun sa stop sequence, and when it reaches the stop sequence, puputol na ngayon yung mismong RNA at hihiwalay na si RNA polymerase. Okay? Tapos mag-merge na ulit silang dalawa. Okay? So yan. Now, pag once nagawa na yung RNA, hindi pa po tapos yan. It has to go processing. So, we have what we call splicing, editing, and also what we call yung polyadenation. Okay? So, let me just go back. So the first one is what we call splicing. So what will happen here is that for it to process, it's going to identify the components of your RNA kasi hindi pa siya complete, hindi pa siya considered as a mature. So i-identify niya as exons and introns. So if you're going to look at the label here on the screen, you will see that some of it is labeled as peach and violet. Okay? Para lang ma-identify nyo. Pero yung iba is exon and as introns. So, what would happen now, once na-identify na niya, it's gonna cut that area, and then you're going to have you're going to have an editing here, which is called the APOB, that actually um, removes the introns, okay, inside the chains. And then, pag didikitin na silang lahat, okay, so you have ribonucleoproteins that will take out the introns. Magkakadikit na si introns, and then afterwards, the last step is the adenylations, okay? So in the polyadenylation, you're going to actually add an adenine base on the terminal end, okay? Because otherwise, it might disintegrate. Okay, so once it's already been adenylated, it's now considered as a mature uh, RNA, mRNA. Okay, so pwede na siya. Ready na siya. Okay? Sorry, hindi ko pala na ano ito yung adenylation. My bad. So yan. Okay, so yan. So it's gonna be adding it. Okay, now the actual three letter sequence that is actually found there is actually based on this information. Para malaman natin which proteins will be called. This is actually called the genetic codon chart. And if you will notice here, it's actually made out of sequence of adenine ACGU, okay, which can be found in your mRNA chain. Okay, so it's actually made of three letters only, okay, and it actually makes your genetic code. Okay, now, so the letters now are combined into what we call segments, or what we call codon. So tatlong letters lang yon, which each can call out for the specific amino acid. So if you look here, sensya na kayo guys ha, may maingit sa paligid eh. So if you will notice here, you have So if you notice here, you have there your different bases. So you have tatlo yan. So you have one here, and isa, dalawa, and then tatlo. Okay? So yan. So there are actually 20 common amino acids that can be generated, and there are 6 possible codons that are enough to produce the 20 amino acids. Kaya nga, if you will notice, since masyadong marami yung sequences na pwede nating i-produce, some of them are actually repeating. So if you will notice here, CUU to CUG is actually leucine. Ito pa, UUA to UUG is also leucine. Diba? So if you will notice, some of them are multiple. Okay? Yan. So nito, puro ALA. To VAL. Ito, threonine. So if you will notice, all of them are your non-essential proteins. Okay, now, with the start of, let's have an example of translating the codon. So let's have the UCA. So you have here the first letter, which is U. So lahat to puro U. They all start with letter U. Itong part na to. Then the second one is letter C. So ito yung column ng letter C. So pag tugmain natin yon, you have apat na UC, di ba? And the last one is A. Now, based on this one, if you're going to look at it, wait, no. If you're going to zoom this out, yan. Ito yung UCA. So, let me just move this a little bit. Yan. So, you have U, C, and A. And it's actually calling for the amino acid serine. Okay? So, yan po yan. Okay? So, take note that the specific row where all the three intersect contains the amino acid that we are looking for. So that is serine. So let's have an example here. Okay. So if you're going to look at the PowerPoint, so you have there CCG. So anybody could give me the mRNA codons ng CCG? Doloberges. Asaya.
GGC po.
Yes, that's good. That's GGC. Wait lang ha. Control. I cannot control. Yes, it's GGC. Very good. And that would be what amino acid? Okay. So if you recall here, GGC, GGC. So ito yon. That's glycine. Okay? So that is actually going to be glycine. So later on, actually you will have this kind of activity. Okay, so isa sa assignment nyo yan. So you're going to fill it up and then identify the amino acid. Okay, so what yung isang assignment nyo is gagawa kayo ng panibagong DNA, and then yung DNA kayo gagawa kayo ng codons, mRNA codons, and then get the amino acid. So for dito, so it's glycine, then GCT is gonna be CGA. Remember, it's already mRNA, so therefore, hindi na po siya magiging A pa rin siya, pero hindi siya magiging U pag nag-back tayo. And then, this is equivalent to arginine. Okay? So, how about for this one, AGC? What would be the amino acid? Sayang na naman. Anybody else? Okay, Miss Medina.
TCG po ba, ma'am?
Ha? Remember, A is no longer, thymine is no longer available in RNA. It's gonna be
Ah,
U. So, ano na siya?
U,
U, ano na siya?
UCG po.
Okay, so it's gonna be UCG. And UCG, as I mentioned earlier, it's gonna call for serine. Okay? How about this one? GTC. Saya na naman. Abarientos.
CAG po.
CAG. Okay, and this is actually gonna call for glutamine. And then last is TGA. Kanyas.
ACU po, ma'am.
Okay, very good. That's going to be ACU. And ACU is equal to reunion. Okay? So, yan. So, yan. Nagawin nyo rin yan mamaya. Kasi marami akong binigay na activity sa inyo, guys. Pasensya na, ha? Na-enjoy tayo gumawa eh. Pero actually, madali lang to. Once you get the hang of it, this is actually fun. And then, there are also what we call mutations. When you see mutations, these are actually, the sequences are actually going to be altered. Okay? So, we have what we call, there are cases when we have, carcinogens, mutagens that could affect the DNA sequence, lalo na kung pumasok siya or naka-apekto siya sa cellular level. Okay, okay lang sana kung magiging X-Men kayo, maggaganon. Alam mo yun, mag-change lang kayo ng ano nyo or you have the power of the mind. But unfortunately, hindi ganun na nangyayari. Malay nyo, di ba? Na-expose sa mutagen yung inyong mother while you're still pregnant. Tapos, ang nangyari, instead of having a soft, straight hair, kinky hair ang nangyari sa inyo, di ba? O kaya instead of having the nose ng tatay ninyo, hala, naging sarat yung itsura ng ilong nyo, di ba? So, yon. So, those are actually examples of mutations or side effects of the mutations. Nag-alter kasi yung protein sequence. And when the protein sequences are altered, it's actually give you a different characteristics, okay? Yung gene expression na tinatawag. Okay? So the first one is what we call a frame shift mutation. So given here that you have your original DNA, UUA, okay? So ang nangyari is, it's supposed to call for leucine. So what happened is that na-alter yung inyong sequence, nakadelete ka ng tatlong base pairs or dalawang base pair, so naiba tuloy yung sequence mo. So guys, pinakita lang tong isa, ibig sabihin this is part of another base pair. Dapat sa kabilang codon siya. Since nagkaroon ng mutagen, natanggal yung dalawa, kaya na-move siya forward. So once it moves forward, nagbago na ngayon yung base pair o yung amino acid na tinatawag. So instead of calling for leucine, it's gonna call for ano ba yung UUG? Balikan natin. UUG. UUG. Okay, it's still calling for leucine. Okay, swerte lang, leucine pa rin ang tinawag niya. 'Di ba? Para what if it's gonna be giving a different amino acid na. Okay? Next one is the silent mutation. Okay? Yung silent mutation naman, remember as I mentioned earlier, may mga sequence tayo na mararami. Kaya marami sila na minsan yung isang grupo ng first and second codons, pare-pareho sila, mapalitan mo lang yung ikatlo o yung una, walang problema as long as it's gonna call the same amino acid. Okay? So, napalitan yung C, pero may UNA. Swerte yung C pala, CUA is still gonna call leucine. Diba? That's what we call silent mutation. Okay? Now, the other one is what we call the missense mutation. When you say missense, pinalitan mo yung first base pair niya, first letter ng codon, it actually called for another um, amino acid. Okay? So, yan yung missense. So it will fall a different kumbaga imbes na iba yung days na tawagin, nag-iba tuloy siya. Then the other one is the nonsense mutation. Pag sinabi mong nonsense, ayun, nagbago ulit siya, pinilitan yung second, o pwede siyang first and second, pero kasi yung UAA guys is actually what we call the stop codon. If you recall the table here, okay, you have there three base pairs for stop. Okay? Yan po. Ayan o. You have three base pair for stop. UAA, UAG, and UGA. So yun, nag-iba lang yung isang letter na stop na siya kaagad. So instead na magkakaroon ka ng long, shiny hair, naging kinky hair. Kasi nga, na-stop yung genetic information mo. Okay? So, yan po. So, those are the different types of mutation. Okay? So, this time, let's go now to the actual translation. Now, before we go to the actual translation, let us identify the different components. Okay? So, unahin mo na. So, you have your tRNA with the bottom as the anticodon at yung amino acid na sa taas. You have your mRNA. Now, dalawang klase po yung mRNA natin. Okay, sorry guys. You have your mRNA, you have the large ribosomal unit and the small ribosomal unit. You have your mRNA, okay, and then remember it's a 5 to 3 prime end. Now, take note that usually when you talk about, before I proceed to the actual steps, yung mismong protein natin, okay, yung mismong transfer, yung rRNA natin has also internal parts, okay? So inside your large ribosomal unit, you have what we call the EPA site, okay? So yung E site is what we call exit site. So dyan lalabas yung transfer tRNA pag natapos na yung trabaho niya. So yung A, this is amino acyl binding site, okay? Or pwedeng sabihin natin accepting site para madali niyo matandaan. And then the P site, ito na yung peptidyl RNA binding site, okay? Or in short terms, polymerization site, okay? Yan, para madali lang tandaan. Pero yun yung talagang specific names niya. And then you have what we call the mRNA binding site. So, dyan siya dadaan. Okay, so yan yung mga parts niya. So, let's start. So, the first thing that happens during the initiation of your protein synthesis, so, dadaan si small mRNA dyan. Hahanapin niya si start codon. Okay? So, hanapin si start codon and it's usually AUG. And AUG is equivalent to methionine. So, tinawag na, so nakatapat na siya doon, si small ribosomal unit. Okay, nakita ko na si AUG, start na tayo, tatawagin niyo na yung transfer RNA. Tapos, saka papasok si large ribosomal unit. Okay, and it starts with the P site. Okay, then what would happen is that it will now move to the next codon. Okay? Hindi ako makita ng magandang ano dito GIF para dito. Pero in-step-step ko na lang. So, move ngayon siya sa next codon. Yung A site will call the next uh, tRNA na tinatawag for that. So, if you're going to, let me zoom in here. Yan. Okay? So, tatawagin na yung next na protein. Then what would happen now is that the proteins is actually going to be transferred from the protein in the pRNA going to the A to the transfer RNA on the A site. Okay? So na-transfer na ngayon siya. The next thing is that magmo-move na yung codon, yung mismong mRNA natin toward para bakante na si A site. So pag sa bakante na si A site, yung kaninang tRNA na wala nang laman, lalabas na siya, mag-exit. Now, continuous yon hanggang sa matapos yung sequence natin. Hanggang sa umabot tayo dun sa stop codon. So, as you can see here on the next slide, I have here a GIF. Ayan, no? So, mag-move siya. Ayan, stop. So, you have here now your stop codon is actually called for the releasing factor that would release your proteins and then end the protein synthesis. Okay? So, yan po. So, here naman is our GIF showing, this is coming from Amoeba Sisters. Kumuha sila ng GIF or somebody made a GIF after their uh, video. Okay. So, yan. This is transcription. Ito naman yung translation. Although, guys, ito nga lang, hindi, medyo may error dito kasi dapat yung large ribosomal unit nyo is big enough to cover all the transport amino acid or your transport protein. Or sorry, the transport RNA. My gosh, sorry naman. So, yan. Sobrang dapat malaki siya. Ito medyo tama yung sa transcription niya. Okay, ito lang. Maliit. Dapat malaki to. Itong peach sa likod. Ayan o. So, paalis sila ng paalis. Okay? So, yan po. Any questions so far, guys? Any question, guys?
No questions.
Okay.
Wala naman po.
Ay, salamat. Okay. Let me just go over some of... Wait lang. Yan. Okay, wait lang guys ha. Buti na lang, malakas ang wifi ngayon dito. Kasi kung hindi, hindi. Wala tayong internet. Okay, so here, ito yung mga dinagdag ko na activity sa inyo. X. Ayan. Ito muna. Okay, so this is the protein activity, DNA to protein activity. So, yan. So, para makita ninyo, let me just open the file para hindi ko na siya download. Wait lang ha. Yan. Okay. So, here, so please do not forget to write your name and the date you finish this. Now, take note is that you will be needing to fill up the necessary parts here. So, anong pangalan nito? Anong pangalan nito? Okay? So, ito na yung given. So, ito po ay summary. Sinama ko siya. Okay? So, ito siguro malamang. This is your... Sige nga. Bigyan natin yung isa. Anong pangalan nito? Anyone? Mariel, Miss Relator, ano pangalan nitong material na to, itong kulay blue?
Helicase po.
Yes, that is helicase. Thank you. Okay, so yan. So at least alam nyo na kung ano yung pangalan niya. Tapos, anong pangalan. Kayo na bahala dyan, sumagot yan. Then, you answer the questions here. Okay, identify the structures based on the description. So, if you want, guys, is sagutan nyo na muna to. Pag na-identify nyo ng parts niya, madali nyo na masagutan to, ma-identify na to. Okay, so yan. And then, number the sequence from 1 to 5. So, 1 is the first one, di ba? So, ano yung first step dyan? Okay, ano yung second, ano yung third, ano yung fourth, ano yung fifth. And for this one, 2 points to, why it is considered as a semi-conservative. Kanina ko pa paulit-ulit na sinusulat yan at sinasabi yan. So, alam niyo na siguro yung sagot dyan. Okay, so ito yung sinasabi ko sa inyo, guys. So, here, let's just remove this. Yan. Okay, so here, so this is the DNA, guys. So the instruction here is fill the complementary DNA strand so both sides of the double helix will be present. Tapos, once na fill up nyo na to ng tamang complementary strand or complementary base, gumawa kayo ngayon ng mRNA. Okay? So, example, di ba? Thymine, anong kapares niya? Angelo. What would be the base pair na kapartner nito? Wala si Angelo? O sige. Diaz, may iba naman. So, Mr. Diaz, what would be the complementary?
Adenine. A.
Adenine. A. Okay, so lagay nyo lang A So, apat na letters lang ang ano nyo. So, pagkasya nyo, since naka-PDF siya, guys, gamitin nyo yung PDF, yung application kung if you're going to use, Kasi po, pag Windows or Office ang pinang-open nyo dyan or Office PDF, hindi ko siya nakikita kaagad pag view. Kaya kailangan ko pa siya i-download. So, yan. So, di ba 8 to, di ba? Sabi ni Mr. Diaz. So, what would be the complementary mRNA niya? Anyone? Ano yung complementary niya? Kung letter A to, ano yung kanyang, pag naging mRNA siya? Sige, Miss Medina.
Uracil po ba, ma'am?
Yes. So, di ba, hindi na siya magiging T kasi nga mRNA na siya, magiging uracil na siya. And if it is uracil, ano ngayon yung kanyang tRNA? Ano yung component niya pa rin? Yes, Isaiah.
Po, uracil.
Ha?
Rin po.
Anong sagot? Sorry. Letter?
Po.
Sorry, hindi ko talaga naririnig, guys. So, nag-fade in ka. Okay, so ulitin natin this is A. This will become U. And then, this will become letter A pa rin. Okay? Kasi yun yung kanyang complementary na base pair. So, since nasa RNA tayo, you follow the thymine. Walang thymine, it's uracil, adenine na sila. Okay? Now, once nabuo mo na tong codon na to, yung codon na to, ito ang gagamitin mo na basis to answer the amino acid sa baba. Okay? So, yan. Let's say it is UGG. So, let's say UGG siya. So, itong gagamitin yung basis. Hindi to yung codon ang basis nyo para basahin dito. Okay? Let's say UGG. Sino ba UGG natin? So, UGG is tryptophan. Okay? So, lalagay nyo dito, TRP. Hindi na po yung buong word na tryptophan. Yung mismong 3-letter words lang. Okay? So, again, ito ang basis nyo para basahin ito. Pero kailangan pa rin makunin nyo yung anti-codon. Okay? Liwanag po yon, guys?
Yes po.
Okay, very good. Okay, so that's the first one. Now, you also have another activity. In-upload ko na rin sa TBL Hub natin. This is again using Bioman. Okay, so nilagay ko siya sa laboratory. Yan. So, again, the same thing ulit. Pagalingan, guys. Mamaya may makita na naman ako mababang score, ha? Na-checkan ko na yung ibang mga ano nyo. Diba si Spider-Man? Huy, alis ka dyan. Yan. Okay. Na-checkan ko na yung iba. Yung final na summary na lang, nakita ko na lahat na nagawa ninyo. Yun ang i-upload ninyo. Okay? Just write your name, your section as well. Okay? So, sasagutan nyo ulit dating gawin, answer the question, and do the game. So, 100 po ito. Okay? So, this is for the first one. The second one is for protein synthesis race. Okay? So, yung protein synthesis race, medyo, ano siya, ano to, magalaw siya. So, pabilisan lang to. Okay? So, yan. Ganun ulit. Answer the questions, related questions, and then screenshot po. Okay? So, both are 100 points. Okay? So, yun po. Any questions so far, guys? Questions? Violent reaction po?
Wala na po.
Wala na. Okay. So, guys, ganito po let me just go ahead and show you. Panoorin niyo yung video ng ano. Malilit lang naman yun eh. Maiksi lang naman. Ha? Wait lang ha. May related video kasi ako dito eh. Sensya na guys, may dito sa school. Okay, ito po. So, this is replication. Mas maganda na nga yung mga videos nila ngayon eh. This is also available in YouTube. Sana mag-ayos siya.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase